Good morning, friends. Good morning, students. And we are here today for another lecture. Now, this lecture will be associated with classification of enzymes, and we'll able to come to know that what are the different classes of enzymes. Since this is one of the important point that is associated in enzymology, because we see a variety of enzymes inside the human body, and all these enzymes have various different roles inside the body. But since their enzymes are said to be functional proteins, their functions are totally different from each other. So their functional variation is always there in an enzyme. But where do these enzymes act? Where are they being classified? Are all enzymes similar or different? What are their site of action? What are their products? All these questions come around when we are learning enzymes. So let us start today's lecture by classification of enzymes. Now classification, in this classification part, we have various different classes of enzymes. So what are the classes of enzymes? Classes are six classes are there in which enzymes are being classified. Oxidative reductases, transferases, hydrolysis, lyases, isomerases, and ligases. Now, if you see the last three alphabets of every class are SES. So this transferases, hydrolysis, lyases. So all SES stands for the enzymes and their particular class. Okay. So the enzymes are being classified into six different classes. Now, these enzymes, classification of their specific coenzymes. Now, how they are being classified? For that purpose, transfer of group other than hydrogen. If hydrogen is not been transferred, then what is been transferred? It is the sugar phosphates, coacid, thymine pyrophosphates, pyridoxal phosphate, folate coenzymes, biotin, cobamide, coenzymes, and lipoic acid. So all these are called as the group transfer mechanisms other than hydrogen. So for a hydrogen, transfer of hydrogen, from where the hydrogen can be transferred is NADP plus or NADPH plus, FMN, FMNH2, FAD, FADH2, lipoic acid, and coenzyme Q. So these are group transfer reactions that takes place. Basically, the hydrogen molecule or hydrogen part of that particular compound gets transferred. One of the example that has been given here, so a glyceraldehyde 3-phosphate dehydrogenase. You can see here, glyceraldehyde 3-phosphate dehydrogenase. So it get transferred into 1,3-bisphosphoglycerate with the help of an hydrogen donation, NADH, NADH plus. Another by group reaction, lactate dehydrogenase enzyme is there, but which enzyme is dependent upon what? It is dependent upon the transfer of hydrogen molecule. So therefore, it will be from pyruvate to lactate with the help of lactate dehydrogenase, and with this reaction of NADH with the addition of H plus and NADH molecules are generated. Now, the system of classification is again based a little bit different. It is called as IUBMB system of classification, International Union of Biochemistry and Molecular Biology, and it was designed in 1964. Again, there were certain modifications that were done in the 1972 and 1978, suggested by IUBMB system of nomenclature of enzymes. It is complex and cumbersome, but it can be clearly understood. As per the system, the name starts with EC, the enzyme commissions, followed by 4 digits. For example, oxidative reductase. What are the type of reaction? The oxidation reduction reaction takes place. Class 2 comes with transferases, transfer of functional groups. Class 3 hydrolysis reactions take place. Class 4 is lyases, group elimination to form double bond. Class 5 isomerases, isomerization reactions. And class 6, ligases, bond formation coupled with ATP hydrolysis. For example, one example they have given how this particular system of classification is ATP glucose phosphotransferate EC2 7.1.1. Now, what this EC2 denotes? Prefix EC denotes enzyme commission. Okay, so here you can see. Just a minute. Here you can see the enzyme commission is here. Okay. Then, which develop the systemic nomenclature. Now, first number 2. This is the first number 2. It indicates the class name. What is the class name? Transferases. Why transferases? See here. Class 2 transferase. Okay. Now, what this transferase is functional for? Transfer of functional group. Now, second number 7. What this 7 indicates? Indicates the subclass phosphotransperases. So, this is the main class. This is the subclass phosphotransperases. Third number indicates 1, which is this one. Indicates subclasses phosphotransperases with hydroxyl group as an acceptor. Now, this was class, this was subclass, and from this subclass, it's subclass, we can say that phosphotransperases, in this phosphotransperases class of enzymes, another hydroxyl group acceptors are there. Therefore, it is classification is 1. Now, fourth number 1, this number 4 indicates D-glucose as phosphoryl group acceptor. Number of particular enzyme in the list. Okay. Now, trivial name is hexokinases. One of the best known enzyme of this particular class is hexokinases. Now, it will be similarly lactate dehydrogenase denoted by 1.1.1.27. Correct. So, 1 will be class, second one will be subclass, first will be this, third one will be subclass, and the last 27th will be, it indicates the acceptor or donation of an electron. This system is set which gives each enzyme a recommended name and a systemic name. An enzyme systemic name is used to prevent ambiguity. For example, malate dehydrogenase, EC number 1.1.1.37 interconverts L-malate and oxaloacetate using nicotinamide adenine dinucleotide NAD as a coenzyme. Its systematic name is L-malate N plus NAD plus oxidoreductase. See here, malate dehydrogenase, what it does, converts malate and oxaloacetate using NAD. So therefore, it is being NAD plus, wherein this NAD will be here, an oxidoreductase reaction takes place, provides a brief chemical description of the reactions. Recommended names. Now, these recommended names are usually formed by adding the suffix. So, what are the suffix? The addition of suffix AAC to the name of the enzyme substrate of the phrases describe a catalytic reactions. Urease, EC 3.5.1.5 catalyzes the hydrolysis of urea. AAC and urea. You can just see here. This is the AAC that has been added and urease. Alcohol dehydrogenase oxidizes alcohol to the corresponding aldehydes. For example, tyrosine, pepsin, and propane are trivial names. Now, class 1, oxidoreductase. Now, this group of enzyme which catalyzes reaction of one substrate with simultaneous reaction of another substrate or coenzyme or transfer of hydrogen or addition of oxygen. A, B, G. You can see here, it is very clearly been donated from here. From this group, the group transfer reaction will take place. Alcohol plus NAD, aldehyde plus NADH plus H plus will be released. The alcohol dehydrogenase, alcohol NAD oxidoreductase. So these are oxidoreduction reaction that takes place by also oxidizing substrate by adding of oxygen molecules. So oxygen molecules get added here. See here. Oxidoreductase reactions takes place. See. Now, what are the classes of enzymes that comes in? Dehydrogenases, oxidases, peroxidases, reductases, monooxygenases, dioxygenases. These are class 1, oxidoreductase. Transferases. This class of enzyme transfer one group other than hydrogen. So this is a particular reaction wherein hydrogen molecule is not very involved from the substrate to another substrate. This may represent as A to R is B represents A plus B into R. So B, the attachment of this R molecule gets transferred to B instead of A. For example, hexose plus ATP, hexose 6-phosphate plus ADP. Correct. So from here, one phosphate molecule will be reduced down and it will be transferred to hexoses. So it becomes hexose 6-phosphate. And since it is one molecule that has been reduced from ATP molecule, this becomes ADP molecule. The name of the enzyme is hexokinase. So kinase AAC, hexoses plus ATP, and systemic that ATP hexose 6-phosphate See here, A, B, and C. A will be left out and B and C will be joined. So it will be transferases, glycosyl transferases, aminotransferases, phosphotransferases. So this is group 2 transferases reaction. Group 3 is called hydrolysis. This class of enzyme can hydrolyze ester, ether, peptide, or glycosidic bond by adding water and then breaking the bond. See here, AB plus H2O gives AH plus BO2. So hydrogen, the water molecule has been broken down. H and OH molecules are being separated. Example is acetylcholine plus water molecule is choline plus acetate. Okay. So this is an enzyme acetylcholine esterase or acetylcholine hydrolysis. These are all basically the digestive type of enzymes come under this class of hydrolysis. For example, esterases, glycosidases, peptidases, amidases. This come under the class of hydrolysis. Fourth class is lyases. These enzymes can remove group from substrate or break bond by mechanism of other than hydrolysis. So the other than hydrolysis, what the reaction may be? Fructose 1,6-bisphosphate aldolysis gives glycerol 3-phosphate plus dihydroxyacetone phosphate. See here, example is lysases, CC lysases, CO lysases, CNCS lysases. So lysases called as synthetase. Enzyme is also called as synthetase. Isomerases. Isomerases kind of reactions that takes place. So these enzymes can produce optical, geometric, or positional isomers of the substrate. Example, recimerase, epimerases, cis-trans isomerases are examples of the same class. Glycerol 3-phosphate get converted into dihydroxyacetone phosphate with the enzyme help of enzyme triose phosphate isomerase. Example of this class, epimerases, cis-trans isomerases, intramolecular transferases. The last class is ligases. These enzymes link two substrate molecules usually with the simultaneous hydrolysis of ATP. Ligate is to bind. For example, acetyl-CoA plus CO2 plus ATP gives malonyl-CoA plus ADP plus PI by the enzyme acetyl-CoA carboxylases. So CC ligases, CO ligases, CN ligases, and CS ligases are classes of this particular kind of enzymes. Correct. So what we have learned into this particular chapter today, we have seen various different classes of enzymes, 6 classes, and 6 classes of oxidoreductase, transferases, hydrolyses, lyases, isomerases, and ligases with various different kind of specificity in their reactions and with their examples. So this is one of the important part that comes usually in the examination also. So this is said to be an important part. Okay. So I need your comments onto this particular lecture today. Do watch this lecture and give me the feedback. So thank you for today's patient listening.